I'll bet you know that insulin is a hormone. You think of insulin as the hormone that lowers blood sugar. The sugar we're talking about, of course, is glucose. What it's really going to be doing for us is not just lowering blood sugar. What we're really interested in is how it's helping blood sugar, glucose, get into the cells. So insulin is a polypeptide hormone. It is not quite large enough to be called a protein, but it is a polypeptide. It's not something you could take orally because this polypeptide would be digested just like a dietary protein. And so the only way really to deliver it is into the blood. Insulin then, the hormone, binds to insulin receptors on its target cells, and that is what triggers the events of this little dotted line here, the mobilization of this hidden away GLUT transport protein out to the plasma membrane and allows glucose to enter the cell. This is insulin, a peptide hormone. It's only 51 amino acids. This is that weird one I showed you before where what is green is carbon, but otherwise everything is the same. And it turns out that this molecule, apparently, they tend to stick together. So in this case, this right here is an insulin monomer. We think that's the active part, but it sticks together with 5 other identical molecules, and the other 5 are the green background here, to form a hexamer. This is the thing, insulin, this part right here is the active peptide hormone that's going to bind to that receptor. This is just a different depiction of the insulin molecule, and each of the little balls that you see over on this side are actual amino acids. And this reminds me of Star Wars or something. Here's the plasma membrane, and these are just 2 different versions of the insulin receptor protein. So this is extracellular fluid here, and this is the intracellular fluid, and there is a portion of this protein which is going to be a site to which insulin will bind. And in fact, that's what's happening here. So the insulin hormone bound to the hormone receptor, and this binding event is going to trigger GLUT4 movement to, I'm going to say PM for plasma membrane. Isn't that enormous? So it has this large portion that sticks outside the cell. Parts of this protein travel through the plasma membrane, and then there's something going on the inside of the cell where this portion of the molecule has to interact with other proteins. And it turns out we don't know what they are, or we don't know what all they are. But this is suspected in what you know as type 2 diabetes mellitus. Okay, that is not how you spell that. Is it 2 L's? Something happens to this receptor, and we become resistant to insulin. And the bet is it's something going on with proteins that interact with the portion of this molecule that is inside the cell. Remember, the point of all of this is to try to get some sort of understanding about the relative size of different molecules and ions that are essential to the operation of a cell. So the hard way to do that is to look at space-filling models of different molecules and try to compare their size, although at the same time it is kind of useful up to a point. Once you begin talking about larger molecules, you're way past that point. So let's take a look at some of the molecules that I gave you space-filling molecules for. I didn't identify it, but there was a methane on one of those slides. Here's its molecular formula. This is actually a much better way to compare than to try to look at different space-filling models. And even better from the molecular formula is to calculate the molecular weight. So I've taken the gases, methane, water, yes, water can be a gas, and I've sorted them according to their molecular weight. And you can see that methane is even smaller than oxygen. Here are probably the most important inorganic ions that we're going to see, the sodium and chloride ions, and they're very small. And then I've grouped the first bunch here. These are the carbohydrates. So glucose has a molecular formula that some of you know, and a molecular weight that turns out to be about half of the molecular weight of sucrose. Sucrose is a disaccharide, so this is a monosaccharide. This is a disaccharide, which by the way is table sugar, which consists of a glucose molecule covalently attached to a fructose molecule, hence the name disaccharide. And if you'll see, it's roughly twice the size, molecular weight of glucose, the monomer. And then here's glycogen, and this is all glucose. It turns out it might have as many as 54,000 molecules of glucose, and it might have a molecular weight of 107,000. That's enormous. I've added in here ATP, and here is the molecular formula of ATP and its molecular weight. So it's larger than sucrose, and at physiological pH it's also charged, and that turns out to be important too. It's harder to move charged things around than if they don't have a charge. For the rest of this, I'm really talking about protein structure. Here's the smallest amino acid, glycine, which has a molecular weight of 75. The largest one, tryptophan, has a molecular weight about 3 times that. And urea, I put urea in here because we have a lab where we talk about urea in AMP1. Urea is a metabolic breakdown product of proteins. I'm not going to talk about metabolism in any great detail, but there are 2 types of metabolic reactions. You know what an anabolic steroid is, right? These are the ones, anabolic, did I get that right? Steroid. Pitiful. These are the steroids, anabolic pathway is what I was trying to write, and I was going to say, hey, as an example, you know about anabolic steroids. Anabolism is that portion of metabolism that deals with building large molecules. So I'm going to say build large from small. And then the other half of metabolism is catabolism. You sort of think of this as digestion. It is breaking down large molecules to small. So when you break down a protein, when you, for example, digest a protein, you have to deal with what we call the nitrogenous waste. Remember, amino acids have that amino group. Urea is the molecule that we use to excrete nitrogenous waste. Look, it's smaller than an amino acid. And then here are our polypeptides. Here's insulin, 51 amino acids. It's got a molecular weight of about 6,000. Here is the glucose transport protein, that thing that has 12 membrane-spanning domains, about 600 amino acids. And here's that enormous insulin receptor protein. Its molecular weight is 156,000 compared to glucose, which is 180, or a sodium ion, which is 23. I'm hoping this is the payoff slide, because remember I told you at the beginning that we were going to be talking a lot about why, how, when, if substances can cross the plasma membrane. If it's a small molecule, particularly if it's a small molecule like CO2 and oxygen, these are non-polar molecules. I think my cats are hunting lizards behind me, so pay no attention to the noise. They go through the membrane on their own. Larger molecules, but not too large, can be transported across the membrane by these very special and very specific transport proteins in a process called facilitated diffusion. And what about insulin? You know, insulin is made by cells in your pancreas. It's a polypeptide. How does it get out of the cell that made it? It can't, it's too large, way large, to go through one of these transport proteins. It's going to leave the cell by exocytosis. Do you remember that? Exocytosis. I think I'm done now.